Hi, good
Hello,
afternoon.
everyone. Oh, go for
Just
it,
wanted to
Leslie.
say
I
hi.
apologize.
Yeah, just wanted to say hi.
Well, thank you, Leslie, for joining us today. Leslie is going to be our second keynote speaker for the Cell and Gene Therapy Virtual Event Series brought to you by LabRoots. The title of her talk today is The Trials and Tribulations of the Clinical Diagnostic Landscape. Before we begin, I'd like to remind everyone that this event is interactive, and we encourage you to participate both by communicating with other attendees using the peer-to-peer chat and by submitting questions during the presentation. Dr. Lesley Northrop is the Chief Diagnostic Officer at Everly Health, and you can visit her complete biography found on your screen. Leslie, you may now begin your presentation. Thank you for being here today.
Great, thanks, Israel. I really appreciate the introduction. Hi, everyone. How you doing today? I don't know where you're checking in from, but it's almost afternoon my time, so I wish you a good day. I really want to thank you for taking the time to listen to my unconventional topic in this more research-focused area. As a former research scientist, my first exposure in coming over to the clinical testing arena was very overwhelming and daunting at times, especially on all the regulations and nomenclature that you had to learn. And so, what I've learned in over 20 years and being on this side of the fence is how to merge the two between R&D and clinical diagnostics. It's really that translational baton pass off between the two that's incredibly important, and that everything starts at ideation, the most fun and interesting part. Otherwise, if you don't start everything at ideation, that baton pass off will feel like a relay race that's in a perpetual loop that will never end, and you'll spend a lot of time and money and risking new innovations coming to market that can change healthcare. So let's dig in. So the talk today is there's 3 key learning objectives that I really would like each of you to be able to take home with you. Maybe some of you already know this and it's just a refresher for you, or maybe some of you are new to this discussion. My goal is really not to bore you with all of the, there we go. My goal is not really to bore you with all this nomenclature, but it's really to engage you and to really understand what it means to get a clinical diagnostic test to market. So these three key areas that we're going to focus on is the whole pipeline of ideation to commercialization. It's a term that I like to use often in my field, and you may have heard it before. But this is really where you need to build your product, a proper product development plan on this pathway from ideation to commercialization of your clinical diagnostic test. So we're going to dig into that. Then we're going to talk about the bread and butter, the validation. This is the most important part of this process. And there's key requirements that you need to be aware of to ensure that you're properly validating your test, and lightly go over what's the difference between an analytical validation and a clinical validation, because they truly have two different meanings. And then, of course, depending on your business, the most important part, right, is long-term viability. It's not about developing your test, validating it appropriately, but getting it into market and making sure that you're able to get the ROI on all that hard work and what you've developed. So we're going to talk a little bit about, getting in-network contracts. Reimbursement is a word that you need to be very familiar with to really obtain that long-term viability. So this is going to be a common slide that you're going to see throughout the presentation. It's a really high-level product development plan, or like we like to say in short, PDP, from ideation to commercialization in a regulated environment. And what I mean by a regulated environment, depending on the type of test that you're validating, as you can see here, that's highlighted with the sun emblem, is whether or not you're going down an LDT path, which is a laboratory developed test path, or an FDA path. I'm not going to dig in on the FDA pre-submission process or the LDT pre-submission process. I'm more focused on the overall architecture of it all. So let's start with research and feasibility. I think what's really important for you to understand here is that you'll see here in the bottom left, I have this question, are you considering reimbursement? This is something that's going to come up commonly throughout the talk, and it's where you're going to get that long-term viability that I discussed. So it's really important that you're thinking about this right at the beginning, where the problem is, and mistakes that most commonly are made, is that people think about it after their test is validated and they're trying to get it commercialized, and then it takes them longer to build that long-term viability. So, the last thing is that you really are thinking about these things at the hypothesis stage, to work on really developing your test, whether or not it's going to be a true diagnostic test or a screening-based test, the test performance of the test. So let's just keep this concept around reimbursement, long-term viability more later in the talk, and let's start at the beginning. So, of course, every great new idea builds innovation. And innovation, I think, for me, is one of the most exciting parts about being a scientist, is really asking questions, trying to come up with answers, and every day something's different, right? Um, but when you're in the ideation stage, in the research and development stage, it's really important for you to ask yourself, what is the clinical unmet need that you're trying to solve? uh, Questions that you can ask yourself: how will it change a physician how they manage an individual's journey to either diagnosing more readily in chronic conditions with overlapping symptoms? Primary example is like endometriosis in women, and this is an example I'll probably bring up many times throughout the talk because it's something near and dear to me. Will it drive an individual to treatment more quickly? Will it lessen unnecessary testing to get to treatment? And will it reduce the healthcare economic burden? That's one of the key areas that our healthcare system is important to understand in order to get that long-term viability for your test. So these are all questions you should ask yourself when you're at ideation in developing your clinical diagnostic test. So, when you're thinking about the clinical unmet need that you're trying to address, here are some key areas just to kind of think tank in your innovation phase is really look at your discovery engine, look at your targets and your markers, high level terminology here that you're trying to associate with a different disease and condition. Are they truly a diagnostic marker, or are they an association with a disease or a condition? There's a key difference between that, and it's important that you understand that at the beginning because that's really going to be how you work on your validation design. You're going to create multiple hypotheses. It's not just about the hypothesis of association of this target or marker with a disease or condition, but it's also about the long term of what this test can do. Is it going to change clinical management? Are you trying to change healthcare, et cetera? Or maybe you're just trying to develop something that's another additional tool in a physician's toolbox to help them better manage their patients. Sorry. So proof of concept. This is also another fun part of the product development pipeline because you're still really in that sort of ideation, research and development phase. And this is really where you're going to start testing under the key components of what an analytical test is. It really allows for you to test at a lower cost before you kick off with the validation. I'm not saying it's always going to be a lower cost, but it's a smaller scale study before you get into the larger validation study. These are some key areas that you really need to focus on when you're in your proof of concept. The sample size, where you're focusing on the indication for testing, which I'll talk about here in a minute. What is indication for testing if you've never heard that before? And determine if the intended use can be met. These are two key areas that we're going to discuss in a minute. It's also important to secure the patient samples or secure samples that are identified in the patient population that you're developing this test for, to really determine the accuracy of the test, the safety of the test, the efficacy of the test. This is where you're going to establish the technology you're going to use. Whether, I'm a molecular geneticist, so a lot of the tests that I've developed is utilizing NGS-based methods or molecular-based techniques. So really identifying which technology is going to get the best answer on what you need to diagnose that condition. It's also important at this part of the stage is to establish the test performance. What does that mean? Well, establish what is the expected sensitivity of your test and specificity of your test and how you're going to achieve it. And then, of course, always determine your test limitations at this part of the phase, whether it's stability of the sample, whether you're going to focus more on a higher sensitivity and a lower specificity, et cetera. So there's a lot of areas here to be thinking about at the proof of concept before you dive into your validation phase. So moving on down the product development plan, let's focus on the product requirements. Let's dig in further on the planning and the development phase around your test product. So this is a term that I just used previously, and I told you that I would talk about it more in detail. This is the most important part, and I can't say that any more uh, greatly in that of any new clinical diagnostic test is crafting your intended use that describes your test, the disease that you're trying to associate with, the performance of your test, deciding is it a true diagnostic test, which we'll dig into further. Is it an aid in diagnosis? Is it a risk assessment? Is it a screening based test? What's the population that you're testing? What's the age group that you're testing? Is it a sex specific based test in only male conditions or female conditions, et cetera? So this is your North Star. And it's really important that you take the time to formulate what your intended use is at this phase before you go into validation. Because when you design and develop your validation, it has to meet your intended use. If you don't do that, you're going to end up having to start over your validation studies, and your hypotheses and your test performance will meet what's expected. So this is just a short-term definition of what an intended use is. What we like to say in the industry, it's either what's the label or the claims on what you intend for your device or test to do. It's more common practice in FDA pre-sub submissions, but as a licensed lab director in the state of New York, this is also another area where intended use is commonly used, and it's really important to start to instill this practice even if you're developing a laboratory developed test. Now, don't confuse indications for testing with intended use. That's something that can commonly occur in that here's a dictionary definition of indications for testing. It indicates the reason for what an individual would use the test for. So it's usually the inclusion criteria for testing of the individual, such as symptoms of the disease or the risk associated with disease. Like I said, it may be an age indication, et cetera. And it's really important to understand what your indications of testing is, because this is something in the clinical testing environment that you usually have on your test requisition form to ensure that physicians are ordering the right tests to meet the needs of diagnosing the condition or the disease. It's also important because this is what is associated if you're going to go for reimbursement. There's that word again. That aligns with the Centers for Medicare Services, or what we call our CPT codes, that you can achieve to get applied to your test so that you can actually have your test covered by insurance. It's not that simple, but indications and testing is a key identifier as part of this process. Now, this is something that I have found in my years of experience, that it's really important to have a really structured, organization around your product development plan. It's probably interesting to hear this in a clinical scientific talk, but it's important that you bring in a portfolio management system or organization. There's a lot of individuals in the field that have experience in this, especially in healthcare and diagnostic. And it's really that this PMO that is the core of your product development plan that's going to really work with all the different areas of your business to ensure, because it's not just the scientists and the clinicians that's part of this process. It could be, the business side. You have your regulatory side, a laboratory operations side, IT, if you have an algorithm based test, et cetera. So it's really important to build out this program in a way to ensure that you have efficiencies through your product development plan, because it'll help you get your test to market more quickly. It'll help you build your decision gates. It'll help you ensure that all the moving pieces and the parts of what you're developing are being accounted for prior to the kickoff of your product development plan. And it also opens up communication, making sure that all the right people and all the right expertise are a part of the conversation while you're in it. I highly recommend establishing this when you're building out your product development plan. So now we're moving on to the, as I said, sort of the gut of your product development plan, your validations. I call this validation and verification. Actually, verifications come before validation, but let's dig into further on these and what each one of those components are, so that you're mindful of this part of the process. So I'm only going to focus on the analytical validity or the analytical design right now. And really what that is this is where you're going to validate or verify your test that it actually measures the specific analyte or targeted marker accurately with reproducible results. It's a very simple definition, and it may be foreign to some of you, but do not confuse analytical validity with clinical validity because they're two different components. What I've listed here, very high level, I'm not going to go into each one and explain each one in detail. That's your homework after this talk. If you don't understand analytical validity design in a regulated environment, you can find it on the CLIA CMS website, College of American Pathology website. There's a lot of areas where you can find this information. What I really want you to understand here is that there's these key areas, and there's other additional areas as well that you need to do, but these are the key areas that you need to focus on when you're validating your tests. The most important thing to notice here is that prior to starting your verification, so verification is usually unblinded, meaning that the samples are not blinded to you. In that you need to make sure that you write up a proper report clearly before you actually start the test performance. The verification is also the step where you pretty much have ironed out your entire protocol, the technology that you're using, and you're doing that one last final step to test it to make sure that it's ready to go into your validation, which is your blinded study. It's also important when you're in this stage in the clinical regulated environment that you have the proper licensed personnel on staff, especially a lab director, before you proceed, because the lab director is the one who's really going to sign off on this, and you need a timestamp that they signed off on it before you actually started the work. As you want to really have in the verification, make sure you have all these kinks worked out before you move to validation, because once you move to validation, technically your test is locked. No changes can be made, right? You're establishing your test performance, you're locking down your assay or your algorithm, whatever your test may be, and then you're putting it through the blinded validation to show that you can get that reproducibility, the precision and consistent test performance, the sensitivity and specificity of your test that you are achieving for accurately to associate that target or that market with a disease condition in the patient population. So I just threw a lot at you here. It can seem very complex if you're new to this, but if your validation does not meet your acceptance criteria as signed off and agreed to prior to running your study, that means you got to go back to verification, you need to modify your SOPs and your processes, and then come back to validation again. So it's really important from ideation to verification that you really hone in on your protocols and make sure that they are meeting all of the expectations that you've established prior going into validation, or you'll waste a lot of time and money. And the most important part about validation, which I have stated, is that it should be blinded, meaning that the personnel that's running the samples in the laboratory to the person who's analyzing the data should be completely blinded to what those samples are and what they're associated with. A lot of times what we use in my business, on my teams, we use what are called honest brokers. So you assign a certain person who holds the key to those samples. And then after you do the analysis and you run the concordance, you then unblind them to see if you actually met the test performance expectations that you established. because you want to remove all bias from your study design. Okay, so now we're going to get into, after following the validations, there's different types of there's different test types. I'm really going to focus on the differences between what is a screening test and a diagnostic test. And I'm using diagnostic tests here pretty lightly. And it depends on who you speak to in my profession. You may get a different answer. However, diagnosis itself is a simple word, right? I mean, it pretty much means what it states, is to diagnose. So now there can be many tools in your toolbox, like I like to say, that incorporates a diagnosis. It's not always a silver bullet. But the rule of thumb here to remember is that if you're developing a diagnostic test, as I've learned in my training and my years of experience, you really want to make sure that that analytical test performance criteria is greater than 95% sensitivity and specificity for it to be considered truly a diagnostic test. Now, some people may challenge me on this because there are diagnostic tests out there that don't meet that criteria, but they're more of an invasive type of test, like a biopsy, etc. And it may be that they have other clinical characteristics or metrics that can be applied to the test to meet that diagnostic definition. But like I said, it's not usually one test, one diagnosis, one treatment. There's other things that are involved in it, but you want to have a really high test performance to meet a diagnostic test. And then screening tests, are screens. That's pretty much what they're known for. They're usually going to have a higher false positive rate because you're screening a more general population. And so there's different things that you're going to focus on your validations in a screening test than you would in a diagnostic test. I think one of the key areas here to see is in point number one. Screening tests usually focus more on the sensitivity because you want to detect that true positive, right? Because you want to drive that individual to a diagnostic endpoint to treatment. Where a diagnostic test, you usually want to focus more on the specificity. It's those true negatives, right? So you want to make sure that that analyte is specific to the disease in question. And usually these two go hand in hand in a lot of cases, but like I said, not every disease and condition follows the same suit because it depends on where we are with science, right? What discovery have we made? Depends on where we are with treatment, clinically, et cetera. So there are some, diagnoses out there for certain diseases and conditions where all you have is a screening test, and there isn't a diagnostic test yet. Where there are some tests that have a really strong clinical care pathway that goes from a screening to a diagnostic. So it's really mindful to really understand what that architecture looks like in the disease and condition that you are actually developing your test for. I think one of the areas where we've commonly can really do a comparison here between a screening test and a diagnostic test is COVID. An everyday person who's not a clinician or a scientist has now truly understand the differences between a screening test, like the fast rapid tests that we can do at home versus a PCR-based test to diagnose a disease. So it's really important to understand the differences here between a screening test and a diagnostic test and where your test falls in to play, because that will help you develop and design your validations. So this slide, I find it's very comprehensive. I'm not going to go through it, but it's pretty much going through all the things I've just stated. It's actually published. It's a published article, and it goes through in detail a really nice assay development pathway through the validation, et cetera, and all the key, what each step is and what is it that you're trying to achieve and answer. So I highly recommend if you're in this phase of your process or you want to learn more about it, the links are here, and I highly recommend that you read up more on it. But what I really want to focus on is some of the caveats to consider in this pathway. There's a lot of things to consider, right? It's a very complex process. The first is, are you actually looking to approach a laboratory developed test or an FDA cleared pre-sub process? That is first and foremost, and especially not unless you were hiding under a rock and you're in this space, you should really familiarize yourself with the FDA ruling and all of the organizations out there that are giving multiple presentations and talks on how to approach this new landscape that we're in, and really, understanding the differences between the two. Also, especially if you are in a state that is a CLIA regulated state, there's differences in how you approach your validations. For instance, if you're in the state of New York or in the state of California, they have a much more rigorous LDT approval process. So it's really important for you to understand what type of test are you looking to develop and validate. As I've talked about, with the product development plan, some of the terminology, I am overlapping FDA terminology with LDT terminology, but the way I've always functioned is that it's important to do both to an extent, depending on, where you are. But it's really important to start building a design control process at ideation. That's why I talked more about this PMO and having these sort of decision gates and controls in place to make sure that each phase of your product development plan, that you're achieving the expectations that are established. It's us samples. I can't, say it more louder from from the rooftops. Samples are probably one of the hardest things to achieve sometimes, depending on the disease or the disorder of the test that you're trying to develop. If you can get clinical samples as part of your validation, that's really powerful. True positives are incredibly important, especially in genetics, the field where I predominantly come from. Establishing an IRB as part of your process so that you could actually run some clinical studies to secure these samples if you can't get them from biobanks or if there are no synthetics out there. It's really important for you to get sample sets that meet the same patient population for that indication of testing for the test that you're developing. Obviously, assay performance is everything. Reproducibility. If your test cannot perform the same every single day, then you've got a problem. You really need to go back to your R&D phase and look into redeveloping the technology of your test. It's also important that anyone in a laboratory environment, that multiple individuals could run the same assay in the lab and get the same performance. So assay reproducibility is incredibly important. As I've said multiple times, blinded validations are important. It's really, key that you make sure that you're removing all bias from your validation design. And then, as I said before, really make sure at the beginning of the design of your test, see if your assay actually meets reimbursement requirements. I am not a reimbursement expert, and I'll say that again here. I have a lot of experience in it. I have key members on my team that are, but it's really important that you have those individuals or have those resources to assess whether or not there's maybe already an established code out there that you could try and get approval for to use for your test, or do you actually have to go and get your own code? It's really particular about the technology of your assay that you're using and the subspecialty of your testing. So this is something to really think about for that long-term viability. All right, so now you're ready to move through the validation, but what if you don't have the established personnel on staff to actually do the validation to meet the regulatory environments? So it's either you have to establish a lab, find a partner that has their CLIA certification that you could validate your tests in, or you need to convert your already research lab into a clinical lab. It's a very daunting task. There's a lot of moving parts and steps that go into it. I'm not going to go into it into this talk, but it's really important that you also start this at the beginning if you are seeking to have a clinical diagnostic test in the market. Like I said, you can't move forward with any tests in the market unless you have a licensed laboratory director. And depending on the state, there's different criteria to meet, but you obviously want to get one that's actually has experience and licensure in the subspecialty of testing that your test meets. Also, staff. You want to make sure you have qualified staff. You have to build a full quality system, around your laboratory operations, as well as technologists, depending on the state. Some states require that technologists be licensed, and some states do not. So it's really important that you understand the state that you're in for the test that you're developing, and what the requirements are to make sure that you're developing your technology under a regulated environment. Okay, so we made it through the long journey from research, ideation, through validation, and you have an established, your test is validated, it met the test performance, you have an intended use, you're really excited. Now you're ready to commercialize it and bring it to market. So let's go over a little bit of that. So what I want to focus on here is not like, okay, your test is out. Now what do you do when your test is commercialized, and how do you get the market, and stuff like that. I want to focus on additional studies that should be conducted either prior to launching and commercializing your test, or immediately following, depending on which regulatory framework that you're under. What I've put here, it's a very high level clinical validity framework, and this is where I'm now stepping out. We've finalized and finished our analytical validity. It's really important that we can also achieve our clinical validity, and I'm not even talking about here clinical utility. Those are other two key areas studies that are important, that are needed to ensure, one, that you get physicians' adoption to utilize your tests, and two, to help with that reimbursement. There's that word again, if you want to get reimbursement. So it's, the process you take your test through, whether it's LDT versus FDA, there's a caveat that you really need to understand here, is if you're not educated on the difference between the two, you should start there. And you should catch yourself up with where we are with this new FDA ruling that is awaiting to, go through Congress and potentially be put into law. I'm not here to talk about that, but it's important to note for you to do your own research. But what I'm going to state is there's another sort of general rule of thumb. The LDTs are garnered, right, under CLIA, and at a minimum, you need to do your analytical validation prior to commercial launching. And it's really important to have some clinical validity, whether it's in the literature, maybe it's an already established association, or to do some clinical studies, or include, like I said, clinical samples as part of your validation. Prior to commercial launch. This is common practice, but I'm not saying it's the best practice, but what I've observed in the industry is most people do the analytical validity, launch their tests, and then we'll follow up with a clinical validation study. So let's just kind of go through the difference here and go through a high level of what this clinical validity framework is. And please note that clinical validation studies are required as part of an FDA pre-sub process and clearance, and your test cannot be commercially launched until that criteria is met. And I'm not going to walk through that right now. So those are just two key differences between an LDT and a FDA cleared test. So some of the key areas I just want to sort of highlight is study design. So it's really important to ensure that there's an established acceptance criteria prior to the start of the study that is well powered. So now you need a statistician on staff based on the prevalence of disease. So you really want to understand the prevalence of disease, because that's going to indicate the clinical sensitivity and the clinical specificity of your test, and also power your study so that you get enough positive samples, in your study design to meet that performance. This is also where you're really going to lean in on the clinical sensitivity and specificity of your test. So like I said, patient population, intervention comparison, and clinical outcomes are really important as part of this process. I've mentioned this previously in the talk, but an IRB, if you don't already have an established IRB process in your institution, I highly recommend it. You also need a clinical scientist, whether it's an MD or PhD, to be the PI of your IRBs. But if you don't know what these are, they're third-party institutions or entities that really make sure they review and approve research involving human subjects to ensure it's ethical and safe. So for your clinical validation, this is where you're actually testing that locked analytical validity of your test in the real world environment, or RWE. So this is where you're actually going to launch it out there in the patient population that meets the indication for testing and see if the sensitivity and specificity of your test holds true. So this is a really important part of the commercialization process of your test. It's also, gathers the data on the safety and the effectiveness of your test. And then clinical relevance of your test. These are key, data points that you need to achieve as part of your clinical validity studies. Positive predictive value, negative predictive value, and here's how you calculate those to really understand how to determine what your positive predictive value is and your negative predictive value is, as well as a false positive rate. These are key terminology that clinicians are going to want to know when you're trying to get them to understand your test and utilize your test in the patient population. All right, we're coming to an end here. So the last thing on the product development pipeline is this long-term viability or clinical ROI. It's really important to understand how to, we like to say, get in network or gain reimbursement. More studies are needed to do that. So clinical validity, which we just lightly talked about, and then, of course, clinical utility. But like I said, a full disclaimer, I am not an expert on this area in a sense that I understand what's needed. I understand the studies and all of that go into it. But there are individuals in the field, RCMs, revenue cycle managers, specialists, who truly understand this landscape and this architecture. And it's important that you have somebody on your staff or a consultant that can walk you through this. And I highly, highly recommend that you start talking about this at ideation, because it can be a very long road, sometimes several years, to achieve reimbursement for your test. And you want to make sure that you're designing your test out of the gates based on the expectations of the payers. So really my only goal is right now is just to get this into your head, that it's important to think about this at ideation. And I'm just going to cover at a very high level some of the key areas to consider. So clinical utility, you've talked about this. Is the test that you designed and developed, is it actually useful? Does it change management? Does it improve healthcare outcomes? Is it show that it's a medical necessity? Does it demonstrate value to payers? And is this test something that, if it does show change, can you get it into clinical guidelines? Because once it's in clinical guidelines, which is really depending on the subspecialty that your test is going to be under, this is physician's guidebook. This is their Bible that they follow on how to treat their patients. Health economic benefits. These are other studies that you can do and publish on. Now, of course, every single one of these studies I've talked about, I've also highly recommend you publish. Publish on your analytical validity, publish on your clinical validity, and publish on your clinical utility. You may have to have multiple clinical utility tests depending on your test design. And health economic benefits. There's millions of papers out there on this in different arenas. So this is really where you want to conduct a budget impact analysis to compare the new test that you've developed and you've got out there to standard of care, and show how you can reduce an annual healthcare cost savings. I have here, if you could click on the example, it's really easy to find more information on this. Also ensure proper coding. So you want to make sure the claims are processed by the payer's administrative system. If there's no existing CPT or PLA code for your test, you may be able to use a general code, or you have to request a new one. It's a lobbyist process. There's systems in place for this. There's meetings in place for this. And that's why I said you need to get an expert on staff to help you with this. And start engaging with payers at ideation. Start having conversations with them. Align your strategy with the payer objective, and demonstrate how a new test supports the desired outcomes for the payers and members. Be proactive and reach out to your payers during ideation. Because if you don't, then you're going to have, if you do it at commercialization, you're looking at a 2+ years, depending, roadmap to get your test into guidelines and payers. All right, so we're going to end the talk here on, I just have some key definitions that I'm not going to walk through, but I highly, highly recommend, if you don't already, you need to know these definitions inside and out. This is around more on the analytical and clinical performance of your test. So I thank you all for walking through this with me. Hope I didn't bore you to death, and I'm welcome to open up a Q&A session.
Awesome. Thank you so much, Lesley, for that informative presentation. Yeah, let's go ahead and dive into the Q&A segment. We encourage you, our audience members, to continue to submit questions for our speaker to answer live in this moment. We already have a few coming in, so thank you for being active in the Ask a Question box there. Let's start with this one here. How do you see the future landscape of clinical diagnostics evolving over the next decade, particularly with advancements in AI and personalized medicine?
That's a great question. It's not actually new to clinical diagnostics. We've been utilizing at least early stages of AI, machine learning, data analytics, et cetera, especially in the genomic space, for quite a while. It's still sort of an uncharted water, I think, from the regulator standpoint on how to manage it, because it's a dynamic process. It needs to be a dynamic process. It cannot be stagnant in nature, because, right, the more data you build into your network and the more nodes that you have, the more precise your test may become, or say if you broaden your patient population, et cetera. I think that you're going to hear more about we're going to regulate this process, because I think people are scared in the fact that it's something, if it's unknown and not tangible to them. It's like believing in Santa Claus, right? You don't see him, but he obviously probably exists in a child's mind. So you have to make sure that your assay, I'm actually giving a talk touching on this a little bit tomorrow at a different meeting. If you're interested, it's on my LinkedIn account. If you want to look it up, I'll dig into it deeper. But you really want to make sure that your assay and your algorithm are working in concert with one another. And right now, your algorithm is a guide to getting to that diagnosis. It's not the endpoint diagnosis, and it's really clear to have that conversation with the regulators about it. But it's here to stay. It's not going anywhere. And I'm really excited about it. And it's something that I'm constantly integrating into my practice and my test development.
Perfect. Thank you for that answer. Let's move on to our next question here. What key components of analytical validity should I focus on?
So I talked about it a little bit. The area that I think is most important is the reproducibility of your test. If you cannot get the same answer on the same sample every time you run the test, then you need to go back and assess the technology that you're using, the sensitivity of that technology. So that's really where, there's all these other factors as well, but to me, that is the most important metric to ensure that your test is accurate.
Understood. Thank you. Let's move on to our next question here. If I am at ideation right now, what is the first thing I should implement as part of my development plan?
Another great question. Obviously, there's a lot of things that you should implement, but I think for the long-term viability of your test and your company, to establish your company and to have it survive in this current landscape, the diagnostic landscape, it's important to understand how you're going to get paid. And you should be thinking about that at ideation, because most tests will launch with out-of-pocket pay, which is great in theory and start, but people really want to, utilize tests that can be billed by their insurance. So I think it's really important to establish that at the beginning to gain that long-term viability.
Great, thank you for that. Looks like we have time for just one more question here. How would you recommend achieving medical payer contracts based on your research?
So as you can see, my disclaimer that I've mentioned multiple times, I didn't go into detail. I didn't walk you guys through the process because it is a very complex process, and I've had some really strong individuals, partners, in the companies that I've worked at, even including here at my current post, that are experts. It's definitely a craft. It's an art. And so I highly recommend that, you reach out to those types of individuals or get access to that information to really understand it if that's something that you're thinking about. The best thing you can do is to bring in a consultant or somebody on staff to help guide you through this process and manage it.
All right. Well, Lesley, thank you so much again for your time and for your important research today. Now, any questions that we didn't have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you registered with. This webcast can be viewed on demand for 1 year until October 16th, 2025. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Thanks again to our audience for joining us today and for your interesting questions. And we encourage you to continue to explore this incredible virtual space while you're here. Until next time, everybody, goodbye. Thank you, Lesley.
Thank you.